Shri Gurubhyo Namaha. We welcome to Online Teaching Academy, Ekalaiva. In the last class, we have studied about the structure of fruit and different types of fruits. Now we will look into seed. Now what is a seed? A seed is fertilized mature ovule. And what does it possess? It possesses an embryonic plant inside it and which stores the food material, and it has a protective coat. So the seed has three parts. That is, if you say that the seed which we are familiar with, we will draw a bean shaped seed. The seed is having a protective coat, and this coat is only what we call it as a seed coat. Seed coat. And inside the seed coat we have two layers. The two layers, what we call it as the outer layer is called as tester, and the inner layer is called as a tegmen. So the seed is having a protective coat which is nothing but the testa and tegmen, and then you find the cotyledon. The cotyledons are there inside this. So the seed coat is the first part. The next one is cotyledon and the embryonal axis. Embryonal axis. So the seed is nothing but it is a fertilized mature ovule, and after fertilization the ovule is transformed into a seed. So a matured ovule, fertilized one, is only what we call it as a seed. And this seed is divided into two different types based on the number of cotyledons, that is dicots and monocots. So you will find dicotyledonous seeds are those seeds which are having two cotyledons, and the monocotyledon seeds are those seeds which are with one cotyledon. And the second type of division of seeds is based on the presence of endosperm. Whether the endosperm is utilized or whether it is persistent, there are two different types are there. So based on the presence or absence of the endosperm, and you all know what is an endosperm. Endosperm is nothing but it is a component which has formed after the fertilization. That is, when you were talking about, we'll recall very quickly, this is the stigma, style and the ovary, and inside this the ovule. The pollen which has fallen on the stigma, it forms a tube, and the male gamete fuses with the egg here, resulting in the formation of a zygote, and other male gamete will fuse with the polar nuclei, resulting in the formation of an endosperm. And this only you call it as triple fusion, you will call it as triple fusion. So the endosperm, whether it is being persistent or it is being complete utilized, based on that we have two different types are there. The first one is what we call it as albuminous seed or endospermous seed, we will call it as. In this, the cotyledons which are there are very thin, membranous seeds, and these mature seeds have endosperm which is persistent. Persistence means it is there till the development takes place. And this endosperm, what is the endosperm has? The endosperm is having a nutritive tissue. So the endosperm is having a nutritive tissue, or you say that it has a reserve food material in it. So this endosperm, which is persistent, will nourishes the seedling during its early development, and such kind of seeds are called as albuminous seeds, you will call it as. So the albuminous seeds will have an endosperm persistent, and this is been nourishing the seedling during the early development. Castor, sunflower, maize are all albuminous seeds. And the second type of seeds are called as ex-albuminous or non-endospermous seeds. In this, the food is utilized by the developing embryo. So, the mature seeds are without endosperm. If the endosperm is persistent, you call it as albuminous. If the endosperm is being utilized, then if it does not have that, then you call it as X-albuminous seeds, you will call it as. In such seeds, you will find that the cotyledons store and become thick and fleshy. And examples of X-albuminous is pea and groundnut. So, the seed is having three parts: the seed coat, the embryonal axis and the cotyledon. And based on the number of cotyledons, we have a dicot and monocot, and depending upon the presence of the endosperm or whether it has been utilized, we have two different types of seeds. One is albuminous and the other one is X-albuminous. Now, when you talk about the dicotyledonous seeds, we said that the dicotyledonous seed is the one which have two cotyledons. As we said that, when you say that the seed is having, if you draw the seed like this, the seed is having, or we will see with this itself, the picture which is there in the page. Here you said that this is the seed coat, and this seed coat is having two layers. What is the layer called as? The outer layer is called as tester, and the inner layer which is present is what we call it as a tegmen. And in the previous slide when we were talking about, we said that the seed is nothing but it is a fertilized mature ovule. So, we will recall a structure of ovule. You said that the ovule is this, having an integuments. You will find an integument here. Now, this integuments results in the formation of the layers, what we call it as tester and tegmen. So, these integuments has formed as a protective coat for the seed, that is the outer tester, the inner tegmen. And when you say that, this is the embryonal axis we are talking about, and these are the two cotyledons which are there. We said that the seed is having three parts: the outer coat, what we call it as the seed coat, and it has an embryonal axis, and the cotyledons are there. Because it is a dicotyledon, it has two cotyledons. And this embryonal axis, what we were talking about, it has a place where the cotyledons are attached. This you call it as cotyledonar node, you call it as. And the above region, this region only is the one which is going to give rise to a shoot system, and we call this as a plumule, you will call it as. And the region which is there below the node, cotyledon node, and which is giving rise to a root system, you call it as a radicle. And the region between this plumule and this node, this region whatever is there, is what we call it as epicotyle, and the region which is present between the node and here below, that is above the radicle, and this region is only what we call it as hypocotyle. So, the seed is having an outer coat, the seed coat, and we will find the embryonal axis which is going to have the upper region. This you call it as, when you talk about this ovule here, this region is called as chalazal end, and this region is called as a micropyle. So, this end, the chalazal end, is going to give rise to the plumule, and this micropyle region is having a radicle which is going to give rise to a root system. And whenever you see that the seed here, this is the micropyle region which is present, and the scar which is present is only what we call it as hilum. Like if you say that this is the ovule, and we said that this is the ovary, and here this ovule is attached to the ovary with a stalk what you call it as funicle. So this is the micropyle. So if you see that the seed which is there, here you will find this is the micropyle and this is the hilum. So hilum is nothing but the scar. Now, so the outermost covering of the seed is the seed coat. So the outermost covering is the seed coat, and this seed coat has two layers. As we said that it has a tester, and the inner layer is what we call it as tegmen. And what are they? They are nothing but the integuments of the ovule. And the hilum is nothing but the hilum which is there. This is nothing but which is a scar on the seed coat through which the developing seeds were attached to the fruit. We said that the ovary will become into a fruit and the ovule is the one which has become into a seed. And above the hilum is a small pole, and that only what we call it as a micropyle. So above the hilum, if you say that this is the scar which you are talking about hilum, here we have a pore, and this pore is only what we call it as a micropyle, you will call it as. And inside this we will find, within the seed we will find the embryo, and this embryo is consisting of an embryonal axis and two cotyledons. So this is an embryonal axis and these two are the cotyledons. And the cotyledons are fleshy, and what do they store? They store the reserve food material for the growing plant. At the two ends of the embryonal axis we find the radicle and the plumule. So you will find this is the embryonal axis, and the upper one is called as a plumule, which give rise to the shoot system, and the other one which is there below this node, what we call it as a radicle, which gives rise to a root system. And in some seeds such as castor, like we were talking about, either it may be endospermous seeds or non-endospermous seeds. So in dicots you will find some seeds are endospermous and some are non-endospermous. So here in castor you will find that the endosperm, which is formed as a result of double fertilization, is a food storing tissue. But whereas in plants such as bean, gram and pea, what is happening? The endosperm is not present because it is being utilized. So it is not present in the mature seeds, and such seeds are called as non-endospermous seed. And we will look into the monocot seed. Now if you talk about a monocot, monocot is having a single cotyledon. So if you talk about the diagram here, you can see this. Here this is the seed, and this is the endosperm, the nutritive tissue which is present, or the tissue which is having the reserve food material, and this is the embryonal axis. Now if you say that here this seed, what is this called as a seed coat? The seed coat and the fruit wall is being fused here in monocot. So you will find that the seed coat, we said that if you say that this is the ovary and this is the ovule, and this ovary will become into a fruit and the ovule will become into a seed. So these two coats are being fused in a monocot. So the seed coat and the fruit wall forms as a single layer here, and in this you will find that two compartments you will find. So this is a larger compartment, and this larger compartment is having a nutritive tissue or the reserve food material, what you call it as an endosperm, and this region will have large amount of food material which is being stored in the form of a starch. And you will find a very thin layer, that is these two compartments are being divided by an epithelial layer here, and here this endosperm is being surrounded or attached to the fruit wall with a very thin layer, and this thin layer is only nothing but an alluron layer. What is this alluron layer consists of? This alluron layer is having a protein content. Proteinaceous material is present in this layer. So the endosperm is having a large amount of reserve food material where we will find starch content in it, and this is the seed coat and the fruit wall which forms as a single layer here, and then you will find this compartment is being separated by a small layer, a proteinaceous layer, what we call it as an aileron layer. And when you see that, here the shield shaped cotyledon which is present, and this shield shaped cotyledon is only what we call it as scutellum, you will call it as. So the shield shaped cotyledon is called as the scutellum, and this is the embryonal axis where you will find that this is the region which is going to give rise to plumule, and this region is the one which is going to give rise to root, what we call it as a radicle. And here in monocot, this plumule is covered by a leafy structure or a sheath like thing, what we call it as coleoptile, and the radicle is being protected or covered by coleorhiza. So these are the three differences what you will find in monocot, that is the shield shaped cotyledon which is present is called as the scutellum, and the plumule is covered by a leaf like structure called as coleoptile, and the radicle is covered by a layer what we call it as coleorhiza, and you will find an aileron layer which is having a protein content, and the endosperm is having starch content in it. So if you see an explanation here, the monocotyledon seeds generally they are endospermic, but some like in orchids you will find that the seeds are non-endospermic, and you will find that in this maize and all, in maize, rice and all, you will find that the seed coat is membranous and generally they are fused with the fruit wall. So you will find that the seed coat and the fruit wall is being fused as a single layer, and the outer covering of the endosperm separates the embryo. So you will find that this is a separative layer which is present, what you call it as an aileron layer, and the embryo is very small and it is situated in groove at one end of the endosperm. This is an endosperm. At one end you will find that the embryo, and it consists of one large shield shaped cotyledon, and this is only what we call it as scutellum, and short axis with a plumule and a radicle, and the plumule and the radicle are enclosed in a sheath. You will find a sheath which is enclosing them. The sheath which is enclosing the plumule you call it as coleoptile, and the sheath which is covering the radicle we call it as coleorhiza. So this is a monocot cell. You have to draw the diagram, and these layers, aileron layer which is protein in content, and the endosperm will have the starch content in it. Now, and a term which we are, when we say that the study of the fruit and cultivation is called as pomology. If you are going to study the structure of seeds, you call it as spermology, we will call it as. Now, before we study the systems of classification, that is three families which we are going to study, we will quickly look into the three different systems of classification. Now, why should we arrange the plants in a systematic order? So that because there are so many diversified forms of plants, and based on their similarities, we have grouped them into various groups. And when you say a group will have similar characters, so that it becomes easier for us to study, and that is what we call it as taxonomy. Taxus is arrangement and normus is rules and principles. Taxus is arranging with certain rules and regulations. There are three different systems of classifications which are there. The first system of classification which was given by Carolus Linnaeus, and that system of classification is called as artificial system of classification. And the second system of classification artificial system of classification which is given by Carolus Linnaeus, who is called as father of taxonomy. And the second system of classification is given by George Bentham and Sir Joseph Dalton Hooker, what we call it as a natural system of classification. And the third system of classification is given by German botanist Adolf Engler and Karl Prantl, and that system of classification is called as phylogenetic system of classification. So in taxonomy there are three systems of classification: artificial system of classification which is given by Linnaeus, and natural system of classification is given by Bentham and Hooker. And phylogenetic system of classification is given by Adolf Engler and Karl Prantl. And this phylogenetic system of classification, Adolf Engler and Karl Prantl, has gone stimulus from the origin of species given by Charles Darwin. And we all know that what is phylogeny, an evolutionary relationship, what we talk about. Based on that, they have given this classification. And natural system of classification is based on direct observation of the plants. Okay, George Bentham and Sir Joseph Dalton Hooker. And this system of classification has been mostly used by most of the Commonwealth countries. We also follow this system of classification. Now, Bentham and Hooker classification, they have divided the seeded plants, that is the angiospermic plants, into three classes. It's called as dicotyledonous. The word itself tells dicotyledonous is the plants which are having two cotyledons, and we'll find that the root system will be a taproot system, the flowers will be pentamerous or tetramerous. So, class 1 is dicotyledonous. The second class is what we call it as a gymnospermy. And what is gymno? Gymno is nothing but the naked, and sperma is seeds. The seeds which are naked have been put under this group called as gymnospermy. And the third class is called as monocotyledonous. That is all the plants which are having only single cotyledon. Because they are monocots, you will find that the fibrous root system will be there and the flowers may be trimerous. So, the seeded plants have been classified into three classes: dicotyledonous, gymnospermy and monocotyledonous. And this dicotyledonous is divided into three subclasses, and the first subclass is called as polypetalae. The first subclass is called as polypetalae. And we have studied what is polypetalae. Poly is free. That is the calyx and corolla are very differentiated, very distinct. All have been put under polypetalae. And the second subclass is called as gamopetalae. What is gamopetalae? If the petals are fused, then you call it as gamopetalae. And the third subclass is called as monoclamidiae. If they have only one whorl, that is undifferentiated calyx and corolla, what we call that whorl as perianth. Then you call it as monoclamidiae. So, dicotyledonous is divided into three subclasses. The first subclass is polypetalae, where they have a distinct calyx and corolla. Then gamopetalae, either they are partially fused or fused, what we call it as gamopetalae. Monoclamidiae, if they are only a single whorl where you don't differentiate calyx and corolla, and such whorl we call it as a perianth. And this polypetalae is again divided into three subseries. The first subseries is called as thalamiflorae. And this series are based on the position of, that is the shape of the thalamus. You will find a dome shaped thalamus, you will find, then they have placed under thalamiflorae. And the second series is what we call it as a disciflorae, a disc shaped, and calyciflorae is a cup shaped. So, the polypetalae is further divided into the series thalamiflorae, Disciflory, and then Calycifloriae. So, based on the shape of the thalamus. What is a thalamus? The receptacle which is going to bear all the floral leaves. So, the series Thalamiflory is further divided into 6 orders and 34 families. Disciflory is divided into 4 and 23, and Calyciflory is divided into 5 orders and 27 families. In this Calyciflory only, we are going to study a family which we call it as Fabaceae. So, the Fabaceae is under Dicotyledone, Polypetalae, Calyciflory. Under this, we are going to study one family, Fabaceae. The second one, when you talk about the Gamopetalae, here you said that the petals are being fused. This is being divided into 3 series. One is called as Inferae, and the second one is called as Heteromerae, and the third series is called as Bicarpellatae. Now, what is Inferae? Inferae is what? Nothing but the ovary is inferior, have been put under series Inferae, and series Heteromerae is if they have more than 2 carpels. Heteromerae, more than 2 carpels if they have put under Heteromerae. And Bicarpellatae is if they have 2 carpels, it is Bicarpellatae. And this Inferae is having 3 orders and 9 families, and Heteromerae is having 3 orders and 12 families, and Bicarpellatae is having 4 and 24 families. And we are going to study one family under Bicarpellatae, that is Solanaceae. We call this family generally as potato family. And under this Monocotyledone, we are going to study one family. That family is called as Liliaceae. So, this is a flow chart of Bentham and Hooker classification. So, we have to know that angiospermic plants have been divided into Dicotyledone, Gymnospermae and Monocotyledone, and Polypetalae, Gamopetalae, Monoclemidae, and we are going to study 3 families in the next class. One is from Fabaceae, the second one from Solanaceae, and the third one is Liliaceae. In the next class, we will see about Fabaceae. Thank you, children. Shri Gurubhyo Namaha.